Good morning. I'm John Button. I'm the lab chief for the Radiation Lab in the Division of Lab Science at CDC, and I'll be presenting on how our lab uses mass spectrometry methods to detect radionuclides. And these methods provide greater response capacity than traditional methods, which is critical in case there were a large-scale contamination event. Then I'll discuss how having quality reference standard materials for radionuclides could improve our capabilities. And so just want to acknowledge some contributors just right away. So Gur, Young, and Olga, they provided information and data about our mass spectrometry methods that they developed. And Jeff leads our elemental analysis lab, and he provided information about their total uranium method. And Robert was the leader of the radiation lab until his retirement last year. And when he started the lab over 20 years ago, his expertise was in inorganic analysis, and he didn't know anything about radiometric techniques. And in my opinion, the success of this lab is really a testament to the synergistic value that you can achieve by combining the two disciplines. And so some disclaimers: I don't have any conflicts of interest to disclose, and all photos in the presentation are by CDC or their stock photos. And the content of the presentation is not official policy by CDC or HHS. So quick overview of the presentation. I will begin with the mission of the CDC Radiation Lab and give an overview of our rapid response clinical lab capacity during radiation emergencies, organized under a framework for lab methods that we call the urine radionuclide screen. And last, I'll discuss how some new standard reference materials specific to the measurement of radioactive materials on mass spec instruments could address certain gaps and lab capacity for rapid response to radiation emergencies. So the CDC Radiation Lab has a mission to provide effective laboratory support for the public health response to exposure to radiologic threat agents. And so what does that mean? Well, radiobioassay is the determination of the kinds and quantities of radionuclides in human samples. And so what is the public need for radiobioassay lab capacity? There are many types of radiation emergencies with different threats and risks to the public. So what will be the demand for radiobioassay during the response to different generalized radiation emergency scenarios? And if we can satisfactorily answer those questions, what does that tell us about the type of analytical techniques that we should develop and validate? In this context, the analytical result does not tell the whole story. A dose assessment needs to be performed. So what do dose assessments and dose action levels tell us about our requirements from analytical methods? Historical radiation emergencies and rad, bio, and chem emergencies, they tell us quite a bit about the potential demands on lab capacity during emergency response. The takeaway here is that in a large-scale emergency, thousands to hundreds of thousands of individuals will require screening for potential external or internal contamination. Luckily, in the large-scale emergencies shown here, the radionuclides spread into the environment that pose the largest risk to human health were strong gamma emitters capable of being detected by gamma portals, thyroid monitors, and handheld Geiger-Müller counters. However, if the radioactive material spread into the environment was a pure beta emitter like strontium-90 or a long-lived actinides, then these types of field screenings for internal contamination may be insufficient. Often in historical emergencies, the number of screened individuals will dwarf the number of truly contaminated individuals by many orders of magnitude. And so I will later explain how our urine radionuclide screen addresses this issue directly. Past emergency responses show how analytical results plus dose or risk assessment can be used in all phases of the response. As the response is beginning or while it is active, results can be used to support critical decisions about how to prioritize the use of scarce medical countermeasures for the treatment of internal contamination or exposure. The results may also be used to monitor the spread of contamination within the population. And as the response enters the recovery phase or after the response is over, results may be used to support creating a registry for affected individuals to support epidemiological studies for the long-term health effects from an acute intake of radioactive material, or to identify at-risk individuals who may require follow-up care or medical screenings in the future. So why were traditional radiobioassay methods insufficient for providing this lab capacity? And what differentiates traditional methods from CDC's rapid response methods? Many of the radiobioassay capable labs are for occupational exposures and contaminations, and the radionuclides that pose a threat to radiation workers can be narrow in scope. For these reasons, the traditional methods had sample throughputs on the order of 10 to 20 samples per day, and they require large sample volumes of urine collected over 24 hours in order to achieve target sensitivities appropriate for occupational exposures. In a public health emergency, time to analytical results for the first 40 samples received is significant because those results may guide critical decisions during the initial phase of a response. 24-hour urine collections are very good for determining exposures near occupational limits, but they would be entirely impractical in a public health emergency. Most significantly, the CDC methods are approved for providing clinical results that guide medical decisions, whereas traditional radiobioassay methods are not. The CDC methods have a high sample throughput using instrumentation dedicated to radiobioassay, but that throughput can be scaled up during a true emergency using ICP-MS instrumentation within our branch of DLS. So as a starting point for data quality objectives for our lab methods, we started with the concept of Clinical Decision Guide, or CDG, which the National Council for Radiation Protection created in NCRP number 161. The CDG is an action level, and it is an effective dose that corresponds to roughly 1% lifetime excess risk for developing cancers or tumors. The effective dose in sieverts is a measure of biological risk, which weights the absorbed dose, joules per kilogram, by impact on the absorbing tissue or organ. And here, lifetime is assumed to be 50 additional years after the exposure in an adult, and then 70 for a child. It's a single quantitative measure useful for communicating to leaders, public health officials, medical doctors, and the public about the risk from acute radiation exposures. In this chart, I've gathered the limits of detection for some of the radionuclides that we measure with methods on various ICP-MS platforms and compared them to the expected radionuclide concentration in spot samples of urine collected on days 1 and 5 post-exposure for intakes that would result in a dose at the CDG action level for children and pregnant women, or CP. This CP-CDG dose is 1/5 the value of the adult dose, in part due to the longer anticipated lifetime of a child or fetus. We target a method sensitivity as limit of detection that is 1/3 the CDG CP day 5 post-exposure value, and we were able to achieve that for the radionuclides shown here. So now I'll transition to the CDC urine radionuclide screen, which is in part our solution to the needs for clinical lab testing in response to a radiation emergency. To provide screening, identification, and quantification results for 22 priority threat radionuclides, the CDC Radiation Lab uses 7 different analytical technologies. The Radiation Lab created the concept of the urine radionuclide screen to deploy methods and analytical technologies in a strategic way. Again, we start with a spot sample of urine of at least 30 milliliters. We prepare aliquots of the sample for analysis by multiple methods if necessary. So this is a 2-layer approach with rapid screening methods on the first layer, which are capable of processing thousands of samples per day. And they search for samples with gross radioactivity levels larger than the normal population background. This is an important capability because historical precedent teaches us that only a small percentage of samples will be contaminated. The second layer of methods are slow throughput, high precision identification and quantitation methods. They are capable of processing hundreds of samples per day, depending on the target radionuclide. Critical to our ability to fill the needs for radiobioassay in response to a radiation emergency are our various mass spectrometry methods. These methods are of varying complexity regarding sample preparation and analytical technique. In total, the methods cover 8 of the 22 priority threat radionuclides that we targeted for method development. 3 of the methods shown here can achieve target sensitivities with a simple dilute and shoot preparation, and with continuing advances in the precision of single quad ICP-MS, it may be feasible to transfer more methods to a dilute and shoot approach. What really makes ICP-MS valuable for us in this context is the sample throughput, shown here as sample measurements in a 24-hour period. On a per instrument basis, the sample throughput is in the hundreds per day, but that throughput is scalable if we surge capacity. For example, for the total uranium method, 6 instruments are currently validated, and the capacity could be scaled up to 19 instruments if we include other instruments within our branch in DLS. Now I will pivot to how metrology of radioactive standards using mass spectrometry may benefit us. So here's a comparison of the method sensitivity for some of the radionuclides we measure on mass spec compared to available NIST SRM radioactivity standards. And of the ones that are available, the CDC Rad Lab has only used the plutonium-239 to complete method development and validation. The radioactive standards referenced here are all several orders of magnitude above the method sensitivities, action levels, and reportable ranges of the methods shown. So according to clinical lab regulations and our division's policies, external proficiency testing or PT programs are preferred. However, except for thorium-232 and elemental uranium, no external programs exist. We run internal PT programs for our methods. However, to grow lab capacity through partnerships with other labs, we will need to create a PT program centered around clinically relevant activity concentrations of likely radionuclides for different scenarios. This is because PT performance is a valuable tool for evaluating partner labs or grant awardees. During method development, validation, and maintenance, use of single radionuclide standards from NIST made in collaboration with the Integrated Consortium of Laboratory Networks, or ICLN, was valuable. The radionuclides and activities were specific to a radioactive dispersal device, RDD, or dirty bomb scenario. And we could foresee a need for similar sets of standards relevant to mass spec techniques for radioanalysis. Historically, participation by the lab in the National Radiochemistry Intercomparison Program, or NRIP, was useful, and the CDC Radiation Lab methods performed well, but it was difficult to maintain participation at NRIP and in our own internal PT programs. So some conclusions. The CDC Radiation Lab answers a unique set of requirements. The lab developed methods using many different analytical instrumentation and techniques. The CDC Radiation Lab was successful at meeting method goals using standards from NIST and from commercial vendors, and the demands for maintaining the current level of capacity are sustainable. But if we grow capacity through lab partnerships and method transfers, we will need to give careful consideration to how we source, create PT samples and standards used for method maintenance. Some additional acknowledgements. I would like to acknowledge our current and past lab staff, as well as our collaborators from US federal labs. And here's a not so recent photo of my team or my colleagues. And please feel free to contact me with comments or questions, specifically like if you would like specifics about our methods. Be happy to share that. Thank you very much for your attention.